Well, previously, I taught you about diffusion and osmosis. Today, we will start with active transport. When a cell membrane it moves molecules or ions uphill against an electrochemical gradient, then the process is called active transport. Now you must be wondering that what is an electrochemical gradient? See, it is the sum of all the diffusion forces which are acting at the membrane, and these forces they could be caused by a concentration difference, or electrical difference, or even a pressure difference. Uh, the active transport it depends on the carrier proteins also, and do not forget that this carrier protein is capable of imparting energy to the transported substance. And in other words, you can say that it has an ATPase activity. All right, now here you can see the comparison of different mechanisms of transport across the cell membrane. Uh, and you can see that both simple diffusion and facilitated diffusion they take place downhill, while the active transport, no matter it is primary or it is secondary, it always takes place uphill. That means from the low concentration to the high concentration. And only simple diffusion is the type of the transport which does not require a carrier, while the rest of the mechanisms they always need a carrier protein. Uh, now, active transport it has two types, and this classification that is based on the source of the energy which is used to affect the transport. In primary active transport, the energy is derived directly from the breakdown of ATP or some other high energy phosphate compound. While in secondary active transport, the energy is derived secondarily from the energy that has been originally created by the primary active transport. Now, the question is that why a primary active transport might be required, because students, uh, you see, large concentrations of certain substances they are required in the intracellular fluid, even though the extracellular fluid it contains a very small concentration of these substances. And similarly, the concentration of certain substances it has to be kept very low inside the cells, even though the extracellular fluid it has their great concentration. Well, the substances which are transported by primary active transport, they are shown here. And the ions, they include sodium, potassium, calcium, hydrogen. And then there are certain waste products and certain nutrients like amino acids and sugars and iron. While the waste product which has to be excreted out of the cell, like uric acid. The mechanism which is underlying the primary active transport that is sodium potassium pump, and mind it that this is also called a sodium potassium ATPase. Now, sodium potassium pump it basically is a process which pumps sodium ions outward through the cell membrane of all the cells, and at the same time, it pumps potassium ions from the outside to the inside. If we look at the components of the sodium potassium pump, then the major component it is a carrier protein, which is a complex of two separate globular proteins, a larger one which is called the alpha subunit and a smaller one called beta subunit. The larger protein it has three specific features that are important for the functioning of the pump. And I will just enlist these three features. You see, number one, it has three receptor sites for the binding of sodium ions on the inside of the cell. As you can see in this figure, because the pump it has to transport the sodium outside. So on the inside, it has three receptor sites for the binding of three sodium ions. And then it has two receptor sites for the binding of potassium ions on the outside. And the inside portion of this protein, it also has the ATPase activity, means it has the enzymatic activity to break down or split the ATP into ADP and energy. Now, if you have to tell the importance of this sodium potassium pump, then you should keep in mind that it controls the cell volume. And in the absence of this pump's working, the cell would swell until it bursts. Because if this pump does not work to expel the sodium out, then the sodium it starts accumulating inside the cell, and along with sodium, greater and greater amount of water that is also retained inside the cell, which increases the size of the cell. It causes the swelling of cell. Ultimately, the cell bursts. Then another important function which this pump serves is by creating an electrical potential across the membrane. And because of this electrogenic nature of the sodium potassium pump, we call this an electrogenic pump. And this electrical potential is so crucial that it makes the nerve and the muscle signals possible. Well, the abnormalities in the number or the function of this sodium potassium pump that is associated with several pathological conditions. I give you an example that the reduction in the number of these sodium potassium pumps in the myocardium, in the heart muscle, that is associated with cardiac failure. And in case of faulty function of sodium potassium pump, there will be excess reabsorption of sodium ion in the renal tubules, and that is associated with hypertension. The amount of energy required that is proportional to the logarithm of the degree that the substance is concentrated. Mm, well, the examples of primary active transport other than sodium potassium pump, that is the primary active transport of calcium ions and that of hydrogen ions. Now, the primary active transport of calcium ions is achieved mainly by two primary active transport calcium pumps, one of which is in the cell membrane and it pumps the calcium to the outside of the cell. While the second pump, it transports the calcium ions into one or more of the intracellular vesicular organelles of the cell, like the sarcoplasmic reticulum of the muscle cells or the mitochondria in all the cells. Well, the other example that is the primary active transport of hydrogen ions. And at two places in the human body, the primary active transport of hydrogen ions that is important. Number one, in the gastric glands of the stomach, and secondly, in the kidneys, especially in the late distal tubules and cortical collecting ducts. Right, moving on to the secondary active transport. In secondary active transport, the energy, it is derived secondarily or indirectly from a gradient which is created originally by the primary active transport. You see that when sodium ions they are transported out of the cells by primary active transport, a high concentration of the sodium ions it develops outside the cell and a very low concentration is inside the cell. Now, this difference, this gradient, it represents a source of energy because the excess sodium outside the cell membrane, it is always trying to diffuse to the interior. And this diffusion energy of the sodium, it can pull other substances along with the sodium through the cell membrane. And this is how the secondary active transport that takes place. Keep in mind that secondary active transport it indirectly uses the energy obtained from the hydrolysis of ATP. Because an already created concentration gradient, it acts as a storehouse of energy for this secondary transport. And a carrier protein simultaneously binds to sodium and another substance. And then the change in the shape of the carrier protein that causes both the substances to cross the membrane at the same time. There are two types of secondary active transport, co-transport and the counter transport. And accordingly, the carrier proteins in these kind of transports, they are the symporters and the antiporters. The symporters they are used in co-transport, while the antiporters, as the name is indicating, this is used in counter transport. Uh, well, you see, the sodium gradient that was already created by the primary active transport. Now, the diffusion energy of the sodium because of the sodium gradient that can bring other substances along with the movement of the sodium, right? Now, if the movement of the other substance that is in the same direction as that of the sodium, then this phenomenon is called co-transport. While in counter-transport, the sodium ions they again attempt to diffuse to the interior of the cell because of their large concentration gradient. However, this time the substance to be transported that is on the inside of the cell and it must be transported to the outside. So you see here that what is the main difference in the primary and secondary active transport? That in primary active transport, there was direct breakage of the ATP at that time, and only one substance that was being transported, say the sodium that is transported from inside to outside, and the potassium that is transported from outside to inside. And because of this transport which is using energy, now another source of energy it has been created in the form of this concentration difference. Now this larger font of the sodium, it is showing that there is a greater concentration of sodium outside while a lower concentration inside. Because of this concentration difference, there is a natural tendency for the sodium to diffuse from the outside to the inside whenever it gets a chance. So now this tendency of diffusing from the outside to the inside, this will serve as a source of energy for secondary active transport. And secondary active transport, it could be a co-transport or a counter transport. So, because of the natural tendency of sodium to move from the outside to the inside, this is serving as a source of energy. So, how do our cells they utilize this source of energy? That at the same time, when the sodium that is moving along a concentration gradient, there is a carrier protein, and this carrier protein has the ability to transport another substance, for example, an X substance, against its concentration gradient. You see, here the concentration outside is already low, and it is greater already inside the cell, but still this substance, say it is a nutrient, it's glucose, it has to be transported from the outside to the inside. So we take the help of a carrier protein, which at the same time binds with sodium and the other substance and transports both the substances in the same direction. And in that case, we call this carrier protein a symport protein. While the opposite phenomenon that takes place in case of the counter transport, here again in counter transport, the other substance other than sodium, it has to be transported against the concentration gradient. You see, there is a lower concentration inside the cell, and the outside it already has a great concentration, but still our cell it has to transport a substance, say a waste product, out of the cell. So again, then it takes help of the secondary active transport, where a concentration gradient of sodium which was created by the primary active transport, this acts as a source of energy. So the carrier protein, when it transports sodium from outside to inside, it uses the energy of the concentration gradient, and at the same time it exports the waste product from the inside to the outside. And here, why do we call it a counter transport? Because the transport of these two substances which are being transported by the by the carrier protein, this transport is taking place in the opposite direction. Sodium is going inside while the other substance it is getting out of the cell. So this is called counter transport, and the carrier protein which is used for this kind of transport, that is called an antipode protein. Uh, the glucose and the amino acids they can be transported into most cells through sodium co-transport. So what happens in the co-transport of glucose with sodium? Um, the transport carrier protein it has two binding sites on its exterior side, one for the sodium and the other for glucose. And a special property of the transport protein, that is that a conformational change, a change in the shape, to allow the sodium movement to the interior, that will not occur until a glucose molecule that also attaches to the carrier protein. So, when they both, sodium and the glucose, they both become attached to the carrier protein, then the conformational change that takes place, and sodium and glucose, they are transported to the inside of the cell at the same time. Now, this is another graphical representation of co-transport of sodium and glucose. Sodium co-transport of the amino acids, that occurs in the same manner as for glucose, except that it uses a different set of the transport proteins. Well, five amino acid transport proteins, they have been identified so far, and each of which that is responsible for transporting one subset of amino acids. And sodium co-transport of glucose and amino acids, that occurs especially through the epithelial cells of intestinal tract and the renal tubules of kidney. So that was about the co-transport. Now we move on to the counter-transport. You see in most of the cells calcium, and in the cells of proximal tubules of kidney, the hydrogen ions, they can be transported out of the cells through the sodium countertransport mechanism. If we look at the sodium countertransport of calcium, so that occurs in the proximal tubules of kidney, and so what happens there?